Good morning to Dr. Chung and everyone. My name is Yugastiran Alaki and today I will be presenting my research proposal poster titled Isolation of the alginate lice producing Bacillus subtilis from the Sarawak forest soil for biofilm degradation of the multi-drug resistant Pseudomonas aeruginosa. Let me start with the introduction of this study. The antimicrobial resistance is a growing global health concern, and one of the most problematic pathogen that is causing this is the multi-drug resistant Pseudomonas aeruginosa. This bacterium is commonly associated with serious hospital-acquired infection through the contaminated medical devices that can be seen here, and surgical wounds and burn sites. The outbreak of this resistant pathogen has been reported in the Sarawak General Hospital, highlighting the local relevance of the developing an alternative antibiotic strategies. One of the major reasons why Pseudomonas aeruginosa is very difficult to treat is because it can form a biofilm which will protect it from the action of antibiotics. One of the main components of this biofilm is alginate, which is targeted by the alginate lyase, which can degrade and disrupt the biofilm integrity. However, most sources of the alginate lyase that are currently produced are from the marine sources and have limited stability in terms of various environmental fluctuations. Therefore, the study aims to investigate the microorganisms such as Bacillus subtilis that can be isolated from the Sarawak forest soil. And why I choose Bacillus subtilis is because it is considered as well known for secreting extracellular enzyme and also classified as GRAS, meaning generally recognized as safe. Moving on to the objective of this study, the first objective is to isolate the Bacillus subtilis from the Sarawak forest soil. The second objective is to screen and characterize the alginate lyase producing isolates of the Bacillus subtilis. And the third objective is to evaluate the antibiofilm activity against the multi-drug resistant Pseudomonas aeruginosa. Next, I will briefly explain the methods that can be seen here. The first method will be the soil sampling, which will be done from the forest area surrounding the unimass. The soil samples will be diluted and will be grown on a culture medium agar. Then the colonies of that grows on this agar that shows bacillus-like characteristics will be selected and purified. Secondly, the screening for alginate lyase producing bacteria will be done when these colonies grown again on a sodium alginate agar. This formation of the halozone that can be seen here will indicate the activity of the alginate lyase. Third is the identification and characterization of the Bacillus subtilis by using the molecular conformation using 16S rRNA PCR, followed by blast analysis to confirm Bacillus subtilis. The confirmed isolate will then grown and cultured to produce the extracellular alginate lyase, and the enzyme activity will be determined using the DNase assay by measuring the reducing sugar production at 540 nanometers. And then the antibiofilm activity will be evaluated by treating the multi-drug resistant Pseudomonas aeruginosa biofilms in a crystal violet staining process to assess the biofilm degradation. And finally, all experiments will be done in the triplicate and analyzed using the one-way ANOVA followed by the Turkey post-hoc test. Now, moving on to the expected result. What I expect from this study is mainly to produce a highly stable alginate lyase enzyme in both moderately elevated temperature condition and also other environmental fluctuation, potentially showing better stability compared to some marine-derived alginate lyses. Furthermore, I also expect the enzyme to effectively degrade the multi-drug resistant Pseudomonas aeruginosa biofilms. It can be seen here in the figure 2, you can see the crystal violet staining. I expect a lighter crystal violet staining because it indicates the reduced biofilm biomass after the alginate lyase treatment. These expected results will then support the hypothesis that the alginate lyase can weaken the protective biofilm matrix by degrading the alginate. In conclusion, I hope this study may contribute towards the development of alternative and environmentally sustainable approaches for controlling biofilm-associated antimicrobial resistance. That's all from me. Thank you for your attention.